So for those of you who don't know me, I'm John Glass at the JCVI. For those of you who have cells, it's either me or Kim Wise that has sent them to you. Um, and what the reason you are here today is for um, our first ever, let's see, wait, um, I'm working on it. Many of you went to so are you seeing my screen now?
Yes.
So today is our minimal cell workshop, day one. And the reason I have asked so many of you here is that our community keeps growing. But for the last 20 years or so, and maybe even before that, I'd been thinking about that along with some of my colleagues at the JCVI. And but the idea of a minimal cell is not something that we came up with. So as early as the 1930s, um, there were groups. Max Delbruck, in the 1930s, his phage school was thinking about a minimal cell. I couldn't resist. I asked my daughter to make it. I hope you appreciate her efforts. But as early as the 1930s, people have been thinking about a minimal cell as a way to investigate the basic principles of cellular life. And it is a common theme. So I looked in in PubMed yesterday, and if you search for minimal bacterial cell, you can see there are almost 10,000 publications. And I'm responsible for a few of them, some of them even before I joined the JCVI. And I think many of you in the audience probably are responsible for some of these. But in 2016, after almost 20 years of thinking about it, the JCVI finally produced this minimal cell that many of us are working with, JCVI Syn 3.0 and derivatives, but it was a long road to get here. When the Institute started, I'm going to move this over here. When the Institute started, then as TIGER, the Institute for Genomic Research, so the idea was that you could use DNA sequencing to get at what life was about. And they sequenced the first organisms. Clyde Hutchison and Scott Peterson did the first global transposon mutagenesis right before the year 2000. And then what became the synthetic biology department at the J. Craig Venter Institute, but was then the Institute for Biological Energy Alternatives. So Hans Smith and Clyde Hutchison and Craig and others developed a rapid method for whole viral genome sequence synthesis. And then we went on and developed all of these techniques in a series of papers in Science, all of which were aimed at eventually getting us to this minimal cell that we would design and build by synthesis so that we could use this to investigate the first principles of cellular life. And one of the things when we published this in 2016 that I really didn't anticipate happening was this idea that, let's see, gotta move my slides along. Sorry, bear with me. Is that so many people would write us and call me right after this paper published because they wanted to use our cell and work with us to develop this idea of investigate or to investigate the first principles of cellular life. And so in the 5 years since we published, our consortium, if you look at this map, we are, well, I won't say that we're spread uniformly across the globe, but there are more than 45 organizations, mostly academic, a few commercial, that are using the minimal cell to try and investigate basic principles of cellular life, to build organisms, to build more complicated organisms, to use this as a chassis on which you can add other things and see how it affects things, but a lot of different purposes. And the research has really picked up. So in the since we published the paper in 2016, there are now, I think in this slide you can see there are 15 published papers using the minimal cell or directly about focusing on the minimal cell. Some of these are computational papers rather than wet science papers, but the pace of activity with the minimal cell has really picked up. I know now that there are, I think, 4 other papers in review, and there are at least another half a dozen papers that I expect to be submitted to various journals before the end of 2021. So the momentum is picking up, and what I had hoped to do today. So for many of you, I'm the node that connects different organizations, different collaborators. And rather than have that, I would like you to get to know each other just a little bit more than you do now. And so this idea today is there are going to be a lot of talks, 35 talks, these short talks on today and a week from Monday. And I want you to, the idea isn't so much to present elaborate seminars, but to give you a flavor of what is going on in other labs. A little bit about why we're using the minimal cell. So we had always thought, these are some of the highlights I think that we're going to get out of this, is that our organism, and we really think that the organism that you will best be able to work with now is not JCVI Syn 3.0, which is a really wimpy organism, but JCVI Syn 3A that has an additional 19 non-essential genes, but it's much easier to pipette, to work with. It divides and looks like a normal cell. JCVI Syn 3.0 is still a useful tool, but Syn 3A is a better tool, I think. But our idea is it's a great tool to investigate the first principles of cellular life. And our vision is that we have a cell where every gene is needed, and we know the biochemical function and role of every gene product. So that when we can get there, we can develop algorithms for genome scale redesign. And in the future, this will enable us to build organisms that devote maximal energy to their purpose, or to redesign microbes for a myriad of different purposes. But in the years since I made the slide that you see here, one of the things you're going to see a couple of talks later today, things that I really hadn't considered. So there are a couple of groups now that we have learned that the minimal cell is incapable of infecting mammalian cells. You can put it on HeLa cells or HEK293 cells, and in a few days, the minimal cell is gone. In 2 weeks, you can't even detect it by PCR. And so researchers are using JCVI Syn 3B, which you'll hear about in the first talk, to investigate pathogenic potential of potential proteins expressed by genetically intractable organisms. And I'm really excited about that. Now, another thing that I think is really cool, and this is a labor of love by myself, Kim Wise, Li Ji Sun, some folks at the JCVI, Elizabeth Tuchalsky at NIST, and James Pelletier, who is now Dr. James, but was originally a physics graduate student at MIT. And we published this in Cell earlier this year, but what we found, so the minimal cell is not a primordial cell, as some people think. It has, but we've accidentally recreated some features of primordial cells. And so the minimal cell, JCVI Syn 3.0, doesn't look like or divide like a modern bacterial cell, or even its Mycoplasma mycoides predecessor. But with the addition of 19 essential, non-essential genes, we have a cell that looks and divides normally. And we identified 7 genes of these 19 that are necessary for the cells to divide via formation of a septum between daughter cells. Now, what I think we've done is we have in SYN 3.0 a cell that looks like cells before the invention of modern cell division. And then with the addition of these 7 genes, you get a cell that divides like a modern cell. I think this is a great platform for future discovery. And then another sort of accidental discovery is that bacterial histone-like protein HU, which is one of the most highly expressed proteins in all bacteria, in almost all bacteria, typically 10,000 or many more copies than that per cell. So when we removed the non-essential gene immediately upstream of the HU gene, we resulted in a drastic drop in HU expression to only 28 copies per cell. So here what we've done is I think we may have accidentally recreated a cell that mimics what cells looked like before the evolution of chromatin. This could be another rich area to explore some things about how cells work. And they're just going to be a whole pile of, if I touch my computer, things go dark for me. This is bad. There are going to be a whole pile of things that I think we're going to get out of this day. And so also after the second session, there will be a discussion section where we're going to talk for an hour or so. And some of the questions that I just wrote down, just a couple, is will it be possible to make the minimal cell a BSL-1 organism? I think we can do that, and I hope we can pull this off. Also, I want to talk about how we can be a more interactive group. I'm going to stop at that. I will say that that for housekeeping, so WebEx expert Tim Staes, he's listed right now as J. Craig Venter Institute, is available. If you have questions, you can contact him via chat. You can address the chat to Tim Staes or the J. Craig Venter Institute or by email. And I will be monitoring time on these talks, and I will let you know when you have talked 9 minutes. With that, I'm going to stop talking, and our first talk is by my JCBI colleague, Andras Cook, and I think he's got a cool story to tell. So I'm going to stop sharing and And so in theory, I should see you all again. I see right now we are up to 66 participants, which I think is super. Andras, the floor is yours. And as soon as you get loaded, the clock will start.